All right. I think we can get started. It is really a great pleasure for me to introduce Professor Mu-Ming Poo. For most of the people in this room, Mu-Ming really does not need an introduction. But today, we have lots of engineers here. So I think I still need to introduce a little bit of Mu-Ming. If I really introduce Mu-Ming, I think Mu-Ming will have no time to talk. So I try to be brief. So Mu-Ming was educated in Tsinghua University in Taiwan, and then went to Johns Hopkins for his PhD. He was a physicist, turned into biophysicist, and then went to Purdue for a brief postdoc. And then he took on faculty positions in many places that I will not repeat. So he laid on his last U.S. post is in UC Berkeley. Mu-Ming, of course, has many titles. National Academy Member of U.S., members of foreign members of Chinese Academy of Science, Academy of. But I wanted to really introduce two things that is particularly important for scientific community in Hong Kong and in China. In Hong Kong, because many of the young students probably don't know, Mu-Ming was the founding head of our life science biology department. So that's really something special. Where we are here now in life science was built by Mu-Ming. And the second thing that starting from 1999, Mu-Ming founded Institute of Neuroscience in Shanghai. Now, this is a formidable neuroscience research institute in global wise. When you talk to neuroscience people, they view Mu-Ming's institute as a really formidable editor. So speaking to the influence of the institute in the field of neuroscience. But I think Mu-Ming's contribution to science in China is go way beyond the neuroscience, because establishment of the Institute of Neuroscience not only established a neuroscience program, but more importantly, it established a new research culture, a culture that in scientific community in China that was badly needed after China opens up, after China starts to invest a huge amount of resources in scientific research. And the internationally recognized research practice, research culture, I think this is something has barriers which far beyond Institute of. Now, Mu-Ming is leading another very important task in scientific community and in global wise, which is the brain science research project. And this, again, will have huge impact, scientific. So this is what I wanted to introduce. Mu-Ming,
Thank you very much, Minjie, for your nice introduction. It's my great pleasure to come back again to present to you a topic now dear to my heart. How do we talk to artificial intelligence people, our brain scientists? Now, I'm willing to talk, and I hope we can initiate more conversations. So I put up this topic for today to discuss with you with my limited understanding of artificial intelligence. And mostly I will tell you what I think is the important problems in brain science and what lesson that might be useful to the design of artificial neural network, new architectures. So let's look at the most hot topic these days, the AlphaGo winning the Master Li. Now, if you ask Master Li to think about which move to make, or a computer to do it, the computer is faster and made the right choice. But if you ask the computer to move the piece to the right position, the Master Li certainly win. He does it quickly, but the computer cannot do it. So they have to ask a human to help. This is not AlphaGo against the AlphaGo plus human against human, right? So let me see this, the artificial intelligence embedded in these robots. The best robot now, one of the best robots, for example.
Simulation robotics is really still in the dark ages. So this is the end of a PhD project from one of the best robotic institutes, and the student has trained this robot to pour this water into a glass. It's a hard problem because the water sloshes about, but it can do it. But it doesn't do it with anything like the agility of a human. Now, if you want this robot to do a different task, that's another 3-year PhD program. There is no...
Now, let's see a human. It's a little girl. Now, what's the difference is that in human brain, they have to do a lot of process, multi-sensory integration. Take a bottle, you have to coordinate your vision, your touch sensation, your movement, everything together. You have do sensory motor transformation quickly. You have to memory recall what you want to do. You have to do motor planning and execution. All this needs to be accomplished by a robot equipped with artificial intelligence. So that's clearly, that's the future. And the future robotics have to go to integrate all these processes in the human brain in order to be as sophisticated. Now, what is in the human brain that makes it different? The cortex. Human brain is enclosed by these thick layers of cells we call the cerebral cortex. This is an evolutionary accident that in the human, some gene mutation created this outgrowth, outblast, or this explosive growth and production of cells in the brain that needs to be accommodated by folding the tissue to get this enough cells in there. This human, in relationship to its closest cousin, the chimpanzee, is 3 times bigger. It is the this cerebral cortex that makes us human. Now, there are other things underneath the subcortical structure, very complicated, but most other mammalian species would have it. So you can study this subcortical structure, but really you want to understand human, you need to know, understand the cerebral cortex and how it controls these subcortical structures. We now know that each cortical region is devoted to different functions. Right? There are motor region, there are somatosensory region, language region, olfactory, hearing, visual, all localized to different brain region. This is known for many, many years, initially by clinicians who noted that if you have a damage of the brain in a particular area, you lost a particular function. Right? So you can now, in modern days, demonstrate this localization of function in the brain by brain imaging in an intact, awake human being. For example, you ask a human a subject to listen to some words. Now, the area that's most active is the back of your brain. It's called visual cortex. Right? This is most active. The other regions also have activity, but no, you can ignore that. The highest activity is on the back. Now, if you want to, ask the subject to listen to a few words, now the hearing, the auditory brain region, the auditory cortex will be light up. They'll be active. This reflects the elevation of the electrical activity accompanied by the use of glucose. You actually monitor radioactive positron emission from glucose. Now, if you want the subject to speak a few words, you have a language area become active. So now you want the subject to close their eyes and do nothing. Just think about a few words. What you get is this. A very complicated activity all across, globally across the brain. So this is what we really want to understand. Over the past decades, we understand the sensory processing in various regions of the brain, but we have a very little understanding about how the brain thinks. What is going on when we think about something or a few words? Right? So this is the challenge. Now, to visualize this challenge or to demonstrate this challenge, I want to give you a very simple example of a very simple animal, model animal, zebrafish. The zebrafish is a small fish, the zebrafish larvae, the young zebrafish. They are transparent, so their brain can be visible if you label the cell with a fluorescence tag or fluorescent marker. So you can actually see what's going on in the brain when the zebrafish is doing something. If we flash the zebrafish on the stage, immobilize the head with a piece of alga, a gel, and allow their tail to move. So the flash will trigger escape behavior as reflected by the swinging of the tails. They want to swing away, but they cannot do it because the head is fit. So then we look at what's going on in the brain, right? So this experiment done by Jolin Du in our institute in Shanghai monitor thousands of neurons firing. In fact, he can do tens of thousands. There are probably 100,000 cells in the brain that can be monitored at the same time. But here, the calcium imaging, which monitors the activity of the firing of the cells, you can see this very active activity. Each dot is a neuron. This activity goes on before you even put a flash. So there's a spontaneous activity going on. We don't know what's going on, what is happening in the brain. Maybe the fish is thinking or something, right? Before they react by flipping the tail. Here's another example. This is a bar which gives you a larger flash. Okay, that's the large flash is the bar, a light bar swim across the field. You see that there are evoked response, evoked activity, but there's also a lot of spontaneous resting activity. So that's the challenge to understand. In human brain, you ask a subject go into the brain machine, look at their activity. You'll find that not only their activity driven by their task they are performing, but there's a spontaneous resting state activity reflecting something in the brain that's active. Right? Nobody knows what's going on. This is the mystery in the brain that we need to solve. Now, the complicated activity is produced by a network of cells. There are 10 to 11 nerve cells, or called neurons, of more than 100 cell type. Right? There's maybe hundreds, maybe even thousands of different cell type, each with the different firing properties. And they link together with processes to form this network. Right? We call the neural network, the natural neural network in the brain. But each cell has a basic elements. The three basic elements of a cell is a cell body with processes, cell body with a long, elongated process that sends information out to other cells. We call it axon. They are branch-like, tree branches, like a process we call dendrites. Dendrites are where the cell receives information from other axons, and the junction that the axon meet with the dendrite is called synapse. And synapse is the most critical structure in the brain where the information flow is controlled. Now, one can study this brain structure at 3 levels. If we look at electron microscopy, we can see individual cells. We can see what's going on in the synapse and the structure in the cell, a lot of details. But this will not tell you how the network works. You want to see a large number of cells working together and how the activity goes. If you look at another level, the macroscopic level, we call above millimeter scales, you can actually see activity, you can see structure by, let's say, magnetic resonance imaging. Right? You can use in the proton diffusion in the brain as to figure out where the fiber tracks are. And then those tracks inferred are all these nicely colored bundles of nerve axons. Each bundle of nerve fibers consists of thousands of axons going both ways, to and from the brain regions. And how they work, and the details of their working principles are not clear just looking at this structure. It's two macroscopic. So the real understanding of the brain will come from we call mesoscopic. Now we define mesoscopic level of connections at a micron to a millimeter level. We want to have a micron resolution so we see individual fibers. We also want to have a millimeter resolution so we know where they are projecting to. Their axon usually goes very long distance to other brain region or many centimeters away. So this is the challenge. Now you look at the detail, at every region, the cells are all packed together. It's just complicated jungle you want to figure out. So in order to do this, you really need to label, to have marker of individual cells. Here we colored it. Mark HD cells of different type, and you figure out each cells where they are going and what cells they receive. Now to analyze this complicated network is the real goal of the brain science now. Now in biology, in the 90s, we have this Human Genome Project, which is an international big consortium working together to solve the sequence of DNA in the human. In the year 2000, or after the Genome Project was finished, people said, well, let's sequence let's figure out or analyze all the proteins in the cell. It's called proteomics, Proteome project. That's actually being done now, and in principle, it can be done for many cells. So in the last 10 years or so, people start to talk about the real challenge for biology is connectome. Now the connectome in many different sense. Connectome, you can talk about connectome at the microscopic EM level connectome, which is every synapse you have resolution. You can resolve. Or macroscopic connectome, which you can get from the brain imaging, which people have already done. For many brain imaging studies, give you this crude map of fibers and activities in the brain. But the real challenge of connectome is the mesoscopic connectome, where you have a cellular resolution over a long distance. This is the challenge. Now, the progress has been made, tremendous progress over the past 100 years. I say the last 100 years of brain science is probably equivalent to the 19th century physics. There are progress made, but they are on the verge of having a big breakthrough. At the end of the 19th century, beginning of 20th century, where big breakthroughs are yet to come. This seems to be the situation of the brain science. What we have solved over the last century is how neural signals are coded, transmitted, and stored. And this we know at a cellular level, at a neuron level. For example, you can do a landmark. What are the landmark discovery over the last century? Well, I would just point out 5 landmarks, which are all Nobel Prize winning work. In the early 20th century, we figured out that the unit of the information transfer in the brain are pulses of electricity. Called action potential, impulse. This impulse lasts 1 millisecond long, fixed amplitude. The information is coded by the frequency and the timing where they started. Right? So this is how the information is coded in the brain, not by the amplitude of the electrical signal, but by the frequency and timing. This is we know since early last century. We also know how this pulse was generated. What generated is actually not the electron, but ions. Ion movement in the nerve cells that gives this pulse of potential change in the nerve cell that can propagate along the nerve fiber, right? With a speed much smaller, much lower than light in the speed of light. It's not much slower than the speed of electron. It's move at hundreds of meters per second. It's still pretty fast at the speed of sound. Right? We also know how this signal reaches the synapse, that they can transfer the signal to the next cell. Now the transfer is not directly electricity goes to the next cell, or the ions goes to the next cell, but you have to exchange through the exchange of chemical signal secreted by the presynaptic cell to a postsynaptic cell by the chemical exchange that triggers electricity in the next cell. And that's how the exchange of information. So this is slow. So if you have a network that function many synapses, then each synapse is slowed down by 5 to 10 milliseconds. So if you have a long range, when you see a car, you want to put your brake, right? That takes time, right? That's tens of milliseconds minimum, because there are many synapses before my sensory stimulus transmitted through several synapses to reach my motor action. Right? So that synaptic delay is a limitation of the natural network. There's a reason for this, because this complicated structure allows a lot of things to happen, a lot of plasticity, changes of the efficiency of the synapse, which is very important. But we also know very clearly many cells in different parts of the brain are feature detectors. Now, in the visual system, there are some cells detect a spot, a line, or line of different orientation, or an edge in the visual system. That's the feature detectors is the coding of the cell for particular features. Right? This is happening first discovered in the visual system, but now in more higher level perception of the space or location of an animal in different part of space. It turns out they are space cells. Well, some cells will fire when the animal is in a particular location. Another cell will fire when the animal is in different locations. So this is like a GPS system, right? Of space, the space sensation or space localization signal. This was actually the prize given 2 years ago to a Nobel Prize. Now, we know at a cellular level how information are coded, are transmitted and stored, but we don't know how that signal comes by. We know a cell would fire when the animal goes to one place, but why is that cell fire? What is the network that gives the integration of all the information to fire that cell? We know very little, right? So we don't know the neural signal, how the neural signals are processed by local or long-range connections that give rise to the particular cellular firing, neuronal firing in the brain. So with a good understanding of the single cell coding process, but rather poor understanding of the neural network underlies it. And if we talk about how this neural signal generates the sensation, the perception of the animal, the consciousness or the decision making, all this complicated higher function of the brain, we know really very little. Right? So there's a long way to go. And I think it's not an over-exaggeration that we are equivalent to 19th century physics in brain science in terms of the progress of understanding this natural phenomenon embedded in our brain. Now, we think the basic problem is to understand the neural circuit, the subject I want to talk about. What is the structure and the function? Now, so people say, well, let's do brain-like architecture for artificial intelligence, but we don't know how the brain network works. How do we do brain-like simulation, right? European project has a human brain project, want to simulate a human brain with a supercomputer. Now, 95% of the neuroscientists think that's premature because we know so little that your simulation have so many free parameters. You have no way to provide useful information to either brain science or to the AI people. Right? So this is the status. Now, let's just give you an impression how the signal goes in the brain. Let's play a movie, and the green stuff are the blood vessel that provide energy. The signals are transmitted through the cell, generate a signal and propagate over long distance through this axon. And the waves of electricity is waves of ion flow across the membrane, pushes the signal forward. Right? This ionic flow depolarizes the cellular membrane, which is that there's a membrane across, a potential across the membrane. So that signal goes down to the end of the axon. Before it reaches the next cell, it releases chemicals into the cleft, and that chemicals triggers ion flow across ion channels, proteins in the membrane of next cells. This ion could be a positive ion, gives a positive signal, depolarization, or a negative ion gives the opposite signal. So by having this chemical different chemicals secreted, you change the signal from plus to minus. Right? So you greatly increase the power. The inhibitory neuron greatly being in the system, greatly increase the power of processing. Furthermore, this release process, how much chemical to release, how many receptors to activate, the structure of the synapse, all can be changed. So the weight, the synaptic efficacy for signal transmission can be controlled. So you have a control of synaptic weight through this complicated chemical process. And that's endowed the nervous system, the plasticity. In fact, all the memory we keep is at the synapse, billions of synapses in the brain that changes their transmission efficiency after we have the experience. Right? So having a complicated synapse has an evolutionary advantage, endow the nervous system the very important plasticity. Now, how does activity change the structure? Now, the activity we're talking about are sensory, normal sensory activity in the environment, the motor activity we're doing, and the cognitive function we are performing. This experience will create electric pulses in the network. That pulses of currents modify the neuronal or synaptic structure in a permanent way. Or in some cases, a permanent way that gives you learning and memory, right? And once you have the experience, the network is in a different state, and that state gives you different behavior, different cognitive functions, right? So this is what everybody agree. This is what happens. What we don't know for certain is how this activity changes the structure and function. And that's a lot of active research in brain science is focused on this aspect. One of the best idea how the electrical activity can change the structure was proposed by Hebb, a Canadian psychologist, in 1949. He wrote a book, and in this book, he proposed a hypothesis. He said, well, we have all the experience we are receiving. How does that produce a memory, an imprint in the brain? Well, maybe the experiments always cause some correlated firing, simultaneous activation of some nerve cells, and their connection becomes strengthened because of this experience. Once that connection is strengthened, that keeps the memory. That's his idea. Correlated activity, correlated pre- and post-synaptic activity. Two cells fire together, their connection is strengthened or stabilized. Now, the other side of the story is that if they are uncorrelated, if this cell fire, that cell are not fire, then this connection will not be strengthened but weakened. As the opposite. So cells that fire together, wire together. This is a simple Hebb's learning rule. It sounds simple, but it actually has a very important implication. Now, first, is the nervous system really, is synaptic connection really work this way? And in the 70s, 30 years after this proposal, people demonstrated the phenomenon called long-term potentiation, LTP, or long-term depression of synapse. So if we can measure the synaptic strength by the change in the potential in the postsynaptic cells, that amplitude of that potential will certainly increase if you have a high-frequency stimulation of 100 hertz, just for 1 second, 100 pulses at the synapse. The synaptic potential jumps to a new level, high level, and stay there for hours and for days. And so this phenomenon called long-term potentiation. Now, if you give instead of 1 hertz, a low-frequency stimulation for 100 seconds, the same 100 pulses, you produce a reduction of the amplitude, a long-term depression. So the activity, why, according to Hebb's learning rule, this is simple to understand. If you have a high-frequency pulses input, the input gives high frequency, the postsynaptic cell depolarizes. The depolarization, the synaptic potentials adds up. They accumulate and reach a higher level and fire the cell. And so you get a correlated firing between pre- and postsynaptic cell, right? That's fire together, wire together. If you have a low frequency stimulation, this input produces a depolarization that cannot be accumulated because it's still low frequency. You have a small depolarization going on, but they are not able to accumulate this potentiation to fire the postsynaptic cell. So you have presynaptic firing, but postsynaptically they are not firing. So you have an uncorrelated activity fits with the HAPS learning rule. You will be deprived, weakened. So this idea of HAPS is very useful in understanding a lot of things. For example, we can have binding of various visual features through this process of co-activation. For example, if you want to see a particular object in your visual field, each cells or group of cells that are responsive to a particular feature of the visual object, they were activated together, right? So there are cells that responding to the texture of the visual system, to the shape and to the motion, color of different aspects, different features. They are all activated at the same time in the presence of this object. So this object's presence of the object produces a co-activation of this group of cells and produce assembly of cells that are activated. Now you remember your grandmother's face because when you are young, your grandmother's presence always co-activates your group of cells, assemblies in your brain. Right? Your grandmother's hair, her face, her clothes, everything gives you this co-activation of the face feature gives a co-activation of the group of cells. Right? Each actually can represent a group of cells that have been activated by a particular feature, by the nose, by the eyes, etc. And then once this co-activation is repeated substantial times, then you strengthen the connection among them. Then you have the memory. The memory of your grandmother is in your brain. And that cell assembly represents your memory of your grandmother. Whenever a part of the assembly is activated, because of the strengthened connections, you would activate the whole assembly. Your face of your grandmother appear in your brain. So recall is just not a storage like in the computer, storage in a separate place. The storage is embedded in the network, in the synapse. Right? So everything, the experience imprint this network, interconnected cells. Recall is simply a reactivation of this group of cells. This is the HAPS cell assembly idea. That's very, very useful. Now, the HAPS learning rule says that the change in the synapse, when correlated firing appear, you can have a functional change as we demonstrated in LTP and LTD, strengthening or weakening. But it turns out that there's also structural change. You can also change, you can have the structure of the synapse have an enlargement or shrinkage of the synaptic structure to allow more efficient transmission or less efficient transmission. You can even have a formation or elimination of synaptic structure so that you form a new network. Right? The entire network could be changed. Now, this is interesting because if you look at the important signal transmission is carried out by long-range transmission, usually carried by excitatory synapse, now from peripheral to your brain. Different stages is the excitatory signal that's the dominant. There are inhibitory signal helping to shape up this transmission of excitatory signal. But all the excitatory signal is ending on the special structure of the dendrites. I think I jumped. Okay. called the dendritic spine. That on the spine, there's on the dendrite, where the receiving end of the cell, there's a protrusion. This protrusion is called spine. Dendritic spine is where the signal is being received. Now, why such a curious structure? It turns out that the shape of this spine and how efficient is the electrical current or depolarization spread to the main dendrite is regulated by the shape, by the shape of spine. So you have a weight control, the control of the weight of input by the controlling the shape. Right? This has been demonstrated repeatedly in different systems. For example, these spines that you see can become enlarged when you have LTP, right? Long-term potentiation, so that this synapse become more potentiated by enlarging the spines. Some synapses that have LTD, these spines are shrinked, so that the current generated at its tip of the spine will decay much more rapidly than the larger spine. So we actually can demonstrate that not only the shape changes, you can have a formation of new spines. So this is the example in my own lab that shows that when a mice is learning to associate a signal with a field, right? You pair a sound and an electric shock. After pairing, after a while, just giving the sound, the mice would freeze. And that memory, of freezing memory, the field memory, is stored in the auditory cortex by forming new synapses. Now, for example, we recorded in vivo live mice between 3 days after training, field memory learning. There's a new spine appear to form a synapse with a pre-existing presynaptic structure. Or sometimes there are new presynaptic structure form on the pre-existing spines. So you have a synaptic structure already there. You not only can enlarge or cause shrinkage of the spine, you can also form new spine. Any new spine or presynaptic boutons means a new connection is made. Now, once a new connection made, then this particular presynaptic cell, axons, coming from a specific region called amygdala, the field control center. That axon will produce more signal at the auditory cortex. And this formation of new spine and sometimes the elimination of spines is another way where the memory can be stored. Now, I just jump to this I think a very interesting idea. There's a great discovery made 5 years ago by a Caltech, Christopher Koch, a guy who is interested in consciousness in the brain. Christopher Koch found in humans, they made a recording in the hippocampus region storing memory. They found that if they are cells which are specifically responding to Jennifer Aniston. Jennifer Aniston, is a famous movie star. Now, the interesting thing is not just Jennifer's face that's triggered the cell to fire. The name of Jennifer Aniston, her boyfriend's appearance, all can trigger that cells. But if any other signals, any other person or related to other movie star, you cannot trigger the cell to fire. So he called this cell a concept cell, a concept for Jennifer Aniston. Anything associated with Jennifer Aniston will fire this cell. Now, what does this mean? What does this concept form? So I have proposed here a way of using Hebb assembly to represent the concept. Now, I just told you that the grandmother's face is an assembly of a large number of cells which are responding to the eye, nose, all things. That assembly is part of a group of cells that represent the grandma's face. But grandma's concept consists of the story she told me, the song he sung, I have heard, or her name as well, or everything associated with grandmother assembly, representing that aspect of grandmother. this superassembly can be put together by co-activation in the presence of grandmother in my childhood. So during my childhood, a concept for my grandmother is formed because all these cells all these assemblies are linked together by strengthening their connections into this super assembly. So this is the hypothesis. Now, there's no evidence for it, but this seems to me helps ideas a very good way of thinking about concepts. Now, Jennifer Aniston's cell is not a single cell that represents Jennifer Aniston. It's one of these super assembly. Once that cell's firing because the appearance of Jennifer's name fire a group of cells, including the cell I'm recording. And because of the firing of Jennifer's name can trigger the super assembly to be fired, I record Jennifer's concept, including her face or anything related to her, right? So a single cell level, you can find specific responsiveness to particular concepts or features, but that does not represent that the single cell is the cell. But it's part of a large group, now thousands, millions of cells. Now fire together, you happen to record it. So this is how we think about the problem. Neuronal assembly representing different components of a concept are stored in different brain regions. The names stored in the language area, the song grandmothers used to sing is stored in my auditory cortex, everywhere, but they are linked by long-range connections. Now, the formation of this concept depends on correlated firing of the super-assembly of the various brain regions, and the retrieval of the concept is achieved by activation of the assembly. Each represents a different component. Now, plasticity of the brain is so important. That gives us the memory. But it turns out that the plasticity is very limited in our adult brain. Now, we saw this appearance of some synapse, some connections made in the adult brain in the rat, but those are very low frequency events. Large-scale rewiring of the brain does not happen in the brain. The human adult brain is relatively stable. You can find 5–10% at most synapses will change over days and weeks, but they do not rewire. Right? But this, why is it? Now, if we look at the development, there's a huge rewiring. Now, this is the kid after birth, 1 month, 2 months, 2 years. What you see, the changes in the cortex, is largely the formation of new networks, connections. The cell, the number of neurons is almost determined, is fixed after birth. But all that's changing after birth is the wiring, is increased connections. The interesting thing is that there's a tremendous increase of network. This network is much more complicated than the adult network. There are a lot of redundant synapses, non-redundant connectivity made. Why is it? At this time, during this period, the baby has not even learned anything. All the possibilities are open in the brain. So all the wiring connection is made. It's the experience that's going to shape this connection, to make it functional. Now, the reason that the baby is crawling on the floor, couldn't even stand up and walk, because the motor connection is just totally, it's not shaped yet, right? So it's a lot of connection that fire at a way that doesn't give you coordinated movement. But by learning, by playing with the environment, by instruction from the parents, the kids gradually, the baby gradually shape their connectivity to form the real useful network. So if you see the development, this is the monkey brain. The time is sort of compressed. The monkey brain is compressed. So in the early phase of the monkey brain, the number of spines, remember the spines where the synapses are made. The more number of spines, the higher number of the spines you see is the higher number of synapses, right? More connections. So the connections made increasing in the early phase during the first 2 years of the human baby. And then there's this called spine formation or synapse formation, but then there's a drop, a pruning. The pruning is to prune, to to shape up the circuit to give you the right connectivity. And that pruning reduces the connectivity, become consolidated network to be functional. And basically the network is formation process is a way to imprint early experience. It's a memory. The memory is embedded in the structure of the early network. Right? So unlike the mature network, the network is there. If you want to put in more memory in there, you can only make small changes. You can change the shape, change the efficiency, form a few new synapses, but no growth reshaping, pruning, like what's happening in the early days. Right? So this is what I understand. And in fact, so the lesson is that for those of you who has a little baby or will have a little baby in the future, be sure the first 2 years, that's the time. This is important. Now, baby education is more important than anything else for the kid, right? And don't leave it to your 保姆, right? What is a domestic worker? The parents' input is so much important. Okay, so let me continue on the next thing is the new thing that happens in 15 years ago. Now the HAP rule, so the HAP assembly idea is so powerful, but what's missing? Well, the missing thing in the HAP assembly is the timing, is the sequence. When co-activation, we talk about 1, 2, cell 1, cell 2, fire together, 1, 2, 1, 2, 1, 2, this firing. But what about 2, 1? 2, 1, 2, 1 is also correlated. But this is totally different information. 1, 2 information is different from 2, 1, 2, 1, right? But they're all the same correlation. Now, the new revision of this is that the spike timing, the timing of the pre- and postsynaptic firing is important. Correlated pre- and postsynaptic firing can either strengthen or weaken depending on timing. If pre before post is strengthening, if post before pre is weakening. Right? This discovery, in fact, cut off the half of the HAP postulate, which says uncorrelated activity that create weakening. But no, it's not the uncorrelated, but the wrong sequence of the activity that caused the weakening. Right? This is a new revision now known as a spike timing dependent plasticity. And my laboratory made some contribution to this by defining the window. How narrow is the window for this timing? Now, if you're 1, 2, 1, 2, are they 1, 2 correlated? Well, if the correlation, the timing, the window is too long, there's no change in the synapse, either before or after. Only when within 20 milliseconds or so, presynaptic comes in before postsynaptic firing, you get a potentiation. If you reverse the sequence, the postsynaptic cell is already fired, presynaptic cell input comes in, you get a weakening. Now, there's a causality in this because in the pre-post sequence, pre-input actually contribute to the post-excitation. There's a cause. The excitation of the postsynaptic cell is caused by the presynaptic input within 20 milliseconds. So they are rewarded. This is the right contribution, right input, it should be strengthened. If the cell is already fired by other input, and then you come in with an input too late, there's no contribution, it's the wrong input. It should be punished by weakening. So that's a LTD, it's a depression. So you have a causality built into the spike timing dependent plasticity, and that gives you a sequence memory in the brain. Now, for example, now we take this HAP assembly, now we align in a line. Even though in the beginning they are all connected to each other at random, all equal connectivity. If now we give a sequential excitation of this group of cells, right? Pre-post, 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 the downstream synapses are all strengthened because of sequential unidirectional activation. The upstream synapses are all weakened. The opposite on the opposite side. So after this training, you'll get an asymmetric circuit that embedded a sequence information in it. So if you now you have this asymmetric circuit, you can activate the beginning cell assembly that can trigger this sequence information, recall this sequence firing, because the connection downstream has already been strengthened. So partial stimulus now can trigger the entire sequence firing. Just like the gatekeeper for the soccer team, they see this guy, the foot is kicking in one direction, he knows where the ball is going to end. Because he has already in his memory a sequence, much early imprinted sequence. As soon as the initial signals appear, he triggers the entire thing. He knows the end. That set of sequence memories in the brain. Right? So when you test this in the red brain, in fact, it happens. You can put a multi-electrode in the red brain. You give a sequence, a ball rolling across the visual field, firing the cells that are recorded by the electrodes, and you'll find that indeed there's a sequence firing. Each of these circles are the receptive field responding to a particular responding to the light ball. When they are reaching this position, that cell would fire. So the electrode recorded this group of cells. The amount of firing is proportional to the brightness. So you can see that now during the training, you create a sequence firing of this group of cells, right? So you set up an assembly which remembers the sequence firing. Then after the training, you now comes back with a test. You can test it by a flash at the beginning or at the end. Now you test how many sequential firing in the proper direction is being triggered, recalled. You find out there are not more sequence firings recorded by flashing at the beginning side rather than as compared to the end side. Right? So this can be, this is a visualization of actual firing of the cell. In fact, the firing of cell can be, this is a 16 electrode firing randomly when you flash at the starting point before training. During training, you see that these cells are firing in a sequence with time. Right? With a very nice sequence trigger. Now this training, after training, you come back with a flash in the beginning, and then you can see in all of cases you are triggering the sequence firing in the brain. So the sequence memory is stored in the cortex by connections between cells. Right? So now artificial network. So I taught you a lot of things about what's going on in the natural brain. What can we learn from what I told you? Right? Let's look back at the history of the artificial intelligence neural network. Right? Some of the major breakthrough, I think, in the field is in the early 80s, late 70s, early 80s, the introduction of the Hebb learning rule into the artificial network and the development of this backpropagation algorithm. Now, let me say, for those of you who are in the field, I don't need to say this, but for students who doesn't know about this, I'll tell you this, I think, a critical period in the neural network, artificial neural network. Of course, there are latest things you'll know more, the deep brain network, the deep learning structure, multi-layers structure, presumably also learned from the multi-layer signal transmission in the brain. But those are modification, I think a small modification of earlier breakthrough. I call the Hebb learning and backprop algorithm. Now, the John Hopfield, a physicist, in the early 80s, proposed a model. He learned from the brain science that the axon that coming out of a neuron. Now, this is based on early perceptual model proposed, but in the early perceptual model, there's no synaptic modification rules incorporated. Now, Hopfield thinks that when the axon comes out to make synapse with dendrite, this is a dendrite of every cell. They can make dendrites with all the other cells and also make dendrite with the synapse on themselves as well. And so you have a set, assembly of synapses made by a group of N units in the network. Now, he assumed that this synapse can change their weight, plus 1 or minus 1. But the key is that the weight of connections is adjusted by the Hebb learning rule. That pre- and post-synaptic cell, if they are co-active, they will be fired. So by having this rule, the network quickly can adapt a new state. Given an input, and the network can quickly reach a stable state. That stable state is called an attractor state. Now the memory is distributed just like the natural synapse, natural network in all different synapses as well. The memory. The memory is stored in the weights. So and this Hebb field network is the important breakthrough in the field. Then it comes to this layer structure, work done by Rumelhart, Hinton, and Williams. And Hinton's probably played the most major role. It's called backpropagation algorithm. What they did is still many units, but they also have many connections. This connection, these 3 layers are feed forward. Now, one layer, input layer, fits into a hidden layer inside the network. Another group of hundreds of output layers given an output, right? Even each unit given a different output. And this is a network that needs to be trained by teaching. Now, the teacher would ask, when you put a particular input, what the output should look like. If the output doesn't fit with the required output, then the upstream input should be changed their way. If the output is correct, don't change. But if it's error, change. Right? So error backpropagate, backpropagation of error signal. Then that change in upstream synapse is proportional to the amount of the magnitude of error. So this so-called supervised learning is very powerful, and it gives the most powerful. Most amazing design is the design is called a network demonstration, net talk demonstration, which I personally was so impressed that I want to change my study, change my field into this area, because it's so powerful. A simple 3-layer networks with input, hidden output layers, you can learn to talk. By training the network, right? The alphabets, the letters is inputted in different unit, the input unit. The output are sounds, phonemes of the pronunciation of the word. You train a few hundred words, this network started with an error. The sounds like baby talks. It doesn't make sense. It doesn't fit with what you expect. But by tuning the input with the backpropagation algorithm, now the network started to gradually learn to speak. After a while, you put a novel words in, the pronunciation is correct. Right? They learn to talk. Right? So the net talk demonstration is so impressive that Francis Crick wrote an article about it in Nature. Now, Francis Crick, the discoverer of the double helix, the most important theoretician biologist in the field, he looked at this neural network, great excitement. He said, well, especially the backprop algorithm. But in this comment, he said, wow, it's so exciting, but they are not like brain. They are not biological. And he even said it's biologically implausible to have a backpropagation. Because as I said, the neuron has an input at a dendrite, the output at the axon. The information goes from input to output. The cell integrate. But how does the input know what output is correct or not? Now, you use the algorithm, computer algorithm to change it, but in the brain, there's no signal going backwards, no backpropagating signals. So now here is an example that I think the crosstalk between these two areas is so important. I was still young. That was about 20 years ago. I read this. I was excited about neural network. I read the Francis Crick comment, and I thought, well, is it really true that this backpropagation is biologically implausible? People that haven't really looked at a natural network, whether there is a backpropagation, right? Unsupervised self-backpropagation in the network. So we started doing experiments. Right? We said, well, let's form a natural artificial network, or many cells form networks in the culture dish. Now we're going to create synaptic weight change among connections. And then we see whether there's a backpropagation or forward propagation, whatever the changes we create at the synapse. Right? So we produce a four-cell network to begin with, just like the Hopfield scheme. We have a cell, 4 cells with an dendrites and an axon. The axon comes out and makes synapse with all the dendrites, including themselves. Right? So you have many synapses, right? Like Hopfield synapse. So now we say, well, let's create a output, a change in the output. That is create an LTP. So we fire 2 cells from cell 2 to cell 1. That connection, we create a LTP, then the synaptic strength is jump up to a new level. So we have one side that created LTP. This is this synapse, cell 2 to cell 1. Then we ask what happened to all the other synapses, upstream and downstream. And it turns out that there are many other synapses start to change besides the synapse we are looking at. This is where it's induced, but the other synapses also change. So there's a spread. It turns out this spread is backpropagation. Only when you have an output is changing LTP or LTD, the input would go along with it. It will be potentiated or depressed. That's amazing, totally surprising. Right? This is the experiments completely triggered by the neural network, artificial neural network. So we did a very thorough studies because this novel phenomenon, nobody would believe it unless we really characterize it. We spent a lot of time. We first started with LTD, because the error signal is this LTD signal. In fact, it's a motor learning. It's where the backpropagation idea originated in the motor learning, and the math theory of motor learning is the LTD created in the cerebellum that caused the circuit to learn. Right, so we started with the depression. We found that depression does backpropagate. If you have the output have depression, you propagate upstream to the input. Right? It turns out that both LTP and LTD can backpropagate to the input. Not only backpropagate, there's a lateral propagation as well to the other branches made by the presynaptic neuron. There's no forward propagation. So what does it mean? It means that when a cell in a network knows that their output is correct, they will automatically send a signal to tell their input, you made the right input, so that the outputs get potentiated. So why don't you potentiate yourself as well? Right? So there's a signal propagate back in the cell to potentiate the input. If the output is LTD, LTD, I say, well, it's wrong. It's being suppressed. So you better suppress the input as well. So there's LTD backpropagation. So that's great. I mean, it's a totally novel finding. So we wrote this paper for Nature. Right? So this paper broke the record, or 2 records. The first record is the longest article in Nature in Neuroscience. It's 10-page long. I mean, Nature usually gives you 4 or 5 pages is the maximum. They gave 10 pages to describe this phenomenon. Right? It's very important. And all the referees think this is totally novel. Nobody has expected this. That's the first record. The second record is that this is the least cited Nature article in history. Because nobody cited, because what the biologist doesn't know how to deal with this phenomenon. It's not in their thinking. It's not in their conceptual scheme that you have a backpropagation. The AI people never cite it because they never read biology paper. So the end is even today, I'm most proud of this set of experiments. It's the most novel that ever coming out of my lab. But it's still, I think it's still premature because these 2 fields has not fused or not talked to each other. So this finding remain buried, right, in the literature. But no, it's buried in a prominent place if you want to dig it out. Right? Now, the one argument for biologists is that, well, it's great. It's a natural network, but it's in the culture. It's not in the brain. So unless you demonstrate in the brain, we will not believe it. So we spent 10 years demonstrate backpropagation actually happened in the brain. Now, how do you demonstrate it? You demonstrate it in a particular system called retinal tactile system. So here you have a retina that sends input to the tactum. Now, there are many synapses made in retina, several layer synapse. This is light comes in here, then there's several layer synapse, and the output of this retina goes to the brain. Now, this is a serial connection. So our idea is that you can demonstrate the signal created in the brain, LTP and LTD, that happen in the output terminal. If we can see that the input signal in the retina also modified, then we have demonstrated. Right? Now, the reason this system is important is because we know anatomically there's no fibers going from the brain to the retina. Retina is one way. Only retina signal comes to the brain. There's no way that the only way that the brain signal can go to the retina must go through the optic fiber by backpropagation in the cell. Right? That's the idea. Right? So to make a long story short, after 10 years, we have demonstrated this is true, that there's LTP and LTD backpropagate to the synapse. So that's fine. Finally, let me end with all these studies in the natural network. What can be incorporated into the brain? I said the Hopfield and the Backpropagation algorithm is the major breakthrough in the artificial network. These are all small part of the brain, small part of brain science, right? Because there are so many things that I just told you about that we know about the plasticity of brain, etc. So the first thing that one can do for the AI, for the artificial network, is to introduce multiple cell types, multiple neurons. We have the usual artificial neural network is a single type, excitatory. There's no inhibitory, but we know there's many different excitatory, inhibitory neurons. It produces powerful analytical power, add a lot of analytical power to the neural circuit. So that should be incorporated into the neural network. There are multi-directional connections. The connections are not only made forward, the three-layer structures all feed-forward connections. There's no connection back from the output layer directly to the hidden layer and to the input layer, so why not? Right? There's no connection between the cells units within the layer. Why not? Right? We saw that all over the place in the brain. Right? So that's something to add. There are also multiple forms of the plasticity. The Hopfield and Backprop introduced the functional modification of the existing connections. Right? LTP introduced. LTD, I might say. But there are also short-term modification versus long-term modification in the brain. The long-term modification will leave a long-term change in the memory, but a lot of things you don't want to memorize. When you are doing a task, your artificial network might want to forget all of the information, but keep relevant information in your network. Right? The brain does that. Brain has a way of converting short-term memory to the long-term memory when it's necessary. Right? So those are the mechanisms one can introduce. But not only functional efficacy can be changed, structural. Right? Why should we keep the connection always there? You can change the weight, but when the weight goes down to zero, or when the weight reaches the ceiling, why don't you just add a new connections or eliminate that or that weakened connections, just like the brain does, right? So then your network architecture is flexible. It's not a fixed architecture. So that's a structural plasticity. Spike timing. Now the timing is not introduced well into the current most popular network model. It's not only the functionally you have a spike time independence, the structure it can be spike time independent as well. Right? There are backpropagation. Now the backpropagation algorithm gives you error signal to use to modify the input But if you have a correct signal, why don't you use that to strengthen the synapse, right? You can have the propagation not only backwards, also lateral propagation. There's a neuron unit can disappear or newborn. There are new neuron born, neuron disappear. So the number of units, not only connection can change, number of units of the artificial network could change. Why not? Right? If there are situations you think that cells should be eliminated, or why not just eliminate it? All this new architecture would likely to increase efficiency as well as the energy saving. Right? Because the processing, if you have a way of simplify the system, the processing time and energy will be reduced. The rule of conversion of short-term to long-term memory is very clear. There are a lot of rules in the brain we have learned, including the mass learning and space learning. It's different. Mechanism for coding and storage and retrieval and extinction of memory. We have mechanisms, right? So artificial network can learn. There's a reinforcement learning via a different type of cells. Now there's reinforcement learning, as you know, in the AI. But the reinforcement learning are totally different in concept, in their methods from the real brain. That in the real brain, the modulatory, there's a group of units devoted to do reinforcement. This is called neuromodulatory neurons. And their function could be, there could be one additional type put into the network. Besides the excitatory inhibitory, I would recommend a modulated neuron that not only do the modulation rather than signal transmission. Right? There are many other things, for example, the use of nested Hebb assembly. One feature is one nest, one assembly. Assembly put together into super assembly and a super assembly. Those are nested structure. Will be very powerful. There's a topographic organization input. We receive sensory information from the environment. It's all two-dimensional. Right? The input to our network is topographical. Topographical organization, input signals. And finally, the use of synchronous oscillation is very important. This is the timing. The artificial network does not consider timing elements in the network, but having synchronous firing between different groups of units is key to bind features in the brain. Right? So different sensory modality are linked together. We hear the cat's cry and the cat's picture or the visual information, sound and auditory and visual information are always integrated together. And those integration are binding features of long-range binding. It has to be a group of cells in one side of the brain, a group of cells in the other brain. The co-activation binds this information. Right? And the binding could be, co-activation could be phase-locked. Right? One activity before the other. And this phase-lock binding would be key to understanding sequential information, including the recognition of sound, language, and text recognition. So, incorporation of these features one at a time in a simple architecture of artificial AI network, you can review its contribution. Now, I talk to many people in the field, they are not interested. They say, well, our big data set and training of a deep learning network already reached 95%, 97% of accuracy. Why should I add more? As long as I have a big data set, I don't need new architecture. And sometimes they say, okay, they are convinced that let's try one. They just add one additional feature, like spike timing, whatever, into the deep learning network. That's already 97% accurate. They add a small feature into it, it actually drops to 92%. It's bad. No, I told you it's useless. So I'm not going to take more brain inspired architecture. Now, the point is that the brain architecture works not on your totally artificial network, right? So what you need to do is take the simple three-layer structure or hub field network. You introduce one additional feature at a time. Now, the old shallow network, not deep network. Shallow network gives you only 40% of accuracy. You put that additional feature, you might jump to 60%. It's still not as good in your deep learning, big data teaching network, but you save energy 10 times, 100 times, and you can add more. You can increase from 60 to 80 to eventually you have very simple architecture closer to the brain that have a highly efficient energy consumption in getting your output, right? So the approach I would recommend is doing simple network, simple architecture, adding one feature at a time, right? So that's my end of my talk. Thank you.
Yeah, so thank you for a very nice talk. I was just curious, so the backpropagation algorithm typically takes an error function or a loss function that determines how much gets pushed back down. So what would be the physiological equivalence of this?
The physiological equivalence is LTD, a long-term depression created because of the incongruent or wrong timing of the output, right? So that resulted in LTD, long-term depression in the output, And then that long-term depression in the physiology equivalent, what we have shown is that LTD would propagate through the connection itself, inside the cell, to the input, to tune down its input.
But what is it that causes this depression?
The cause I didn't tell you the molecular mechanism. The cause is actually has a molecular basis. The potentiation was done by an axonal transport of a trophic factor called neurotrophin, BDNF, brain-derived neurotrophic factor. We demonstrate that it's a long-term transport, intracellular transport of a protein from the LTP, potentiated synapse, to the input. That's the cause. That factor can modulate the dendritic input by changes receptor concentration, all these things. The LTD is a different molecule, it's a nitric oxide. Nitric oxide triggers signal that goes back to the input as well. And physiologically, it makes sense because when the network is trying to learn, you want to know, without teaching, whether self-learn to change its structure, right? Self-organization. So how do you know the self? How do you know the output? If output is created that doesn't fit with the environmental requirement, it gets depressed. Then you would tell the network that this is the wrong output. Let's tune down the input. The network does itself, not through a computer algorithm, through cellular machinery.
Hi, Professor Poo. So I'm a founder of a robotic company in Shenzhen. We are basically currently embedding artificial intelligence into our robots, and I want to basically find stimulation or things that interest me in bioscience or neuroscience that can do the thing. So you mentioned that deep learning can do, with the big data set, can do certain tasks quite well. But what we are interested in more, we actually facing a real life scenario. So we are building a robot that work in warehouses, and we are facing, it's not thousands, it's like hundreds of millions of different items in a warehouse, and we want the robot to recognize them, to estimate the pose of them, to plan the grasp of them, and plan the motion of how you grasp an item from the shelf to the another robot or to a basket. Right. So the whole thing, we currently using explicit programming as well as deep learning, and the thing is, it cannot scale. Right. So we want the thing we want is transfer learning, basically. So we want
generalization of the neural experience.
Right.
That's what the brain does best.
Right. So
we want a general purpose robot. Right.
to, you're right, I'm building a general purpose robot, and I wanted to basically practice on like a very few examples, just like human, and then extend it to a more general form. And I think the key lies in the structure of the biological neural network. But I want to ask two questions. First is, you have listed 12 different like aspects or features that we might add into artificial intelligence. Do you have like a recommended priority of how to do that? And second,
well,
I don't have.
I'm actually giving the same talk all over the place. All the people who are in the field, they will pick up whatever they want to do. I already the Chen Yunji people in the Institute of 计算所. They pick up the one aspect of it, that's the changing the synaptic connections, allow synaptic to grow or deplete it. They already demonstrated, implemented in hardware. That's highly energy saving. They said 30% of the original required, and timing also much faster. But they are not using the really deep learning, many layer structure, a very simple backprop. Introduce one modification. So I think there are many people can try different aspect to it.
Okay. So actually, my question was actually like, what do you think contributes the most to?
Well, I think the multiple cells.
Okay.
Multiple
type of different.
Introducing our inhibitory neurons.
Okay.
And the connections, rather than feed-forward connections, but feedback. Recurrent natural connections, allowing connection between neurons within the same layer. That's already a big plus. If the connection including negative of inhibitory neuron, I think it will be very powerful.
All right. So my second question is, how much level of neuroscience should I learn before doing that?
You don't need to learn many, but you better talk to a lot of neuroscientists. Your learning is taking a course takes a long time. But talking, a few hours might give you a lot of hints. Right? Then you can direct your reading based on what's the most interesting aspect of it. So I never actually take courses in biology, for example, but I learn through talking to people and learn with finding the interesting thing and study myself. Taking courses is not going to be helpful. But no, we actually are giving courses in neuroscience, in brain science. So we call brain science for AI. It's been given in the Institute of Automation every month, a series of lectures. Those lectures are not normal courses in brain science, but courses to talk about stuff that you AI people will be interested in. That's what I think what we need if we really want to learn deep in the subject. Yes, Prof.
I enjoy your talk very much. I have a couple of naive questions. First of all, for Jennifer Aniston.
Yeah.
When you see and hear and so you from the image you can tell it's her.
Right.
But in large-scale computing, usually we have way to store the data.
Right.
You can, when you call the data, you can form a pattern. But also equally important, you need to find a way to register the pointers, because otherwise you don't know how to call upon certain data. When I hear Jennifer Aniston, I can see mentally her image.
Right.
So that's the other side. So in brain, how do we do that?
Right. So based on, this is all pure hypothesis. Based on the way I think it's working, is that a lot of Jennifer Aniston's cells. There are cells like coding for her face, cells like coding for her name, her sound, her voice, all different areas. So when I recall, part of the cell of this huge super assembly are activated. But because if you have sufficient activation, because once Jennifer will not work, Jennifer Aniston's name has completely appeared, that have produced sufficient activation to wake up all the, through the strengthened connection, all the other related cells in the brain.
My question is, you need a central command to tell which one.
That's the key. the key is that you don't need central command. We don't need a pointer. All we need is to have the same experience that reappear within the same network, right? Then there's no storage site for her name or for her face or area, so we can pick up or recall that. It's all embedded in the network. So when the face comes about, it activates a few units, and that units, because of their learned connections, they would, through the storage, through this activation, directly wake up the entire assembly.
I think the data is distributed, but you still need to know where the data, relevant data are located.
We don't need to know. In terms of the, there may be some segregation, self-organization within artificial network. People are looking into the network to see after you learn, are they specific site where the information are located. But the beauty of this the AI network is that it's all in thousands and thousands of synapses there that embedded all the information. So when the network is working, we don't know how it's actually in the hidden layer where the information are located. But when the letters come in, they just trigger precise connection to give the output a sound, right? So I think the same principle, one could imagine that the embedded information embedded, even it could be discrete area. We don't know how it organized. Well, it'd be interesting to know. But discrete organization might be useful because self-organization would probably achieve that because it's more efficient, right? But all we care is the output actually activates.
That's one side. I'm talking about reverse.
Yeah.
I have the other question in terms of network. Backpropagation is only one of the multiple. There are many different architectures.
Right.
Backpropagation is classical. It's very effective, but it is computationally more intense, more demanding.
Right.
And hence, I would imagine the human brain, since we don't compute as much, as fast, and the energy requirement is larger. They are more localized. I suppose we may have some similar kind of idea. It's just you use somewhat slightly different word than what we use. So if you have different kinds of the radio network, which has a lot more localized data processing than connectivity.
Yeah.
So I'm trying to find some kind of analogy because I'm not an expert in neuroscience. I cannot see how brain will function like a backpropagation machine because that doesn't seem to be most efficient.
Right. So it's just an algorithm. That's one of the feature that brain might use. Like forming a network that you know what's going on in output and you modify the input. It's a helpful way, but it's certainly not the whole thing. Well, the architecture I'm talking about in terms of natural, backward, recurrent connections, or not feed forward but feedback connections. Those are actually what brain uses a lot, rather than backpropagation, what we talk about. Right? So there are other architectures that's involved. Now, here's some statement that I hear a lot, that brain is so efficient. We don't need to learn. We immediately recognize a face or recognize, we learn very fast. But we forgot that the brain has learned throughout the development. All through the life, the circuit has been keep learning countless information, countless teaching, supervised teaching of the network, resulted in what brain we have now. So we are not a naive brain, right? These structures very efficient in using small amount of information to make a decision, right? That's because we have learned tremendously over the past. Right? So to say that the brain is efficient, it doesn't need to be trained, that's probably a little bit misleading. Anyway, I didn't answer your question, but...
So I actually have a more general question. My question is about more into the AI part. So one of the hardest things with the AI is the fact that if we look at the reality, our brain learns by observing things. So we have different senses. And my question is, you were talking about including some features into the shallow learning instead of including a lot of data into the deep learning, which is like easier a little bit. So what kind of features are those, like, are you like thinking about? Are those features related more to the incorporating different senses? Like, because if I want to learn a language, if you show me this bottle and you tell me that this bottle is named like that, I'm going to memorize it and learn it. So what kind of features are you talking about when you talk about, including?
Well, I'm talking about a feature in architecture, not a feature of sensory input. Well, it could be applied to auditory, visual, but one thing that I think one has to a test of capabilities that you can do multi-sensory integration in the network, which are not able to, people are not able to do with pattern recognition, right? You can recognize the sound as well as pattern, and also a motion of the pattern. All this is integrated by the network. Now, these are what I'm saying is that learning this new architecture, if we introduce some aspect, some architectural elements into the network, would help you to develop a network that could do many things like multisensory integration, sequence learning, all these things that could happen. I'm not saying that the network is going to train a particular type of input. I'm talking. Right? See my point?
Got it. Thanks.
All right. So, Mu-Ming, I think you have definitely very vividly and convincingly showed us that modern neuroscience, brain science, is an exciting area for many people, scientists, engineers, to work, and there are many challenging questions ahead. And I think perhaps more importantly, will inspire many young people in the audience to take this on in your future career. With that, I would like to ask you to join me to thank Mu-Ming again for this wonderful talk.
Thank you.